Hello everyone! Today we are starting mass spectrometry instrumentation components. Mass spectrometry is a powerful analytical technique that helps us identify molecules by converting them into measurable information. The complete process starts with the sample. First, the sample is introduced into the instrument. Then it moves to the ion source, where molecules are converted into charged particles called ions. After ionization, these ions enter the mass analyzer. Here, ions are separated according to their mass-to-charge ratio, also written as m/z. Next, the separated ions reach the detector, where their signals are detected and measured. Finally, the data system converts these signals into a graph called a mass spectrum. So, the core concept is simple. A mass spectrometer converts molecules into ions, separates those ions by mass-to-charge ratio, detects their abundance, and displays the result as a mass spectrum. Now, let's understand the complete instrument overview. The mass spectrometer works in 5 main steps. First is the sample. The sample contains the molecules that we want to analyze. Second is the ion source. Here, the molecules are converted into charged ions, because only charged particles can be controlled and measured inside the instrument. Third is the mass analyzer. This is the separation area. It separates ions according to their mass-to-charge ratio, or m/z. Fourth is the detector. The detector receives the separated ions and converts them into electrical signals. Finally, the data system processes these signals and displays them as a mass spectrum. So, from sample input to molecular fingerprint, mass spectrometry gives us detailed information about the identity and abundance of molecules. Now we move to one of the most important parts of mass spectrometry, the sample introduction system and ion source. The sample introduction system is responsible for bringing a small and controlled amount of sample into the instrument. This is very important because too much sample can disturb the analysis, while too little sample may not give a clear signal. The sample can be present in different forms, such as gas, liquid, or solid. For volatile samples, GCMS is commonly used. For liquid samples, biological samples, and less volatile compounds, LCMS is usually more suitable. After entering the instrument, the sample moves toward the ion source. The ion source converts neutral molecules into charged ions. This step is necessary because the mass spectrometer cannot separate neutral molecules directly. It needs charged particles so they can be controlled using electric and magnetic fields. There are different ionization techniques depending on the sample type. Electron ionization, or EI, is a hard ionization technique. It gives strong fragmentation and is useful for small molecules. Chemical ionization, or CI, is softer than EI. It produces less fragmentation and helps in finding molecular ion information. Electrospray ionization, or ESI, is widely used in LCMS. It is excellent for biomolecules, peptides, proteins, and other large or polar compounds. MALDI stands for Matrix-Assisted Laser Desorption Ionization. It is useful for proteins, polymers, and large molecules. So, from sample input to molecular fingerprint, mass spectrometry gives us detailed information about the identity and abundance of molecules. Now this slide shows the complete workflow from ions to molecular information. After ionization, ions enter the mass analyzer. The mass analyzer separates ions according to their mass-to-charge ratio, or m/z. Different analyzers work in different ways. A quadrupole filters ions by allowing only selected ions to pass through a stable path. A TOF analyzer separates ions by their flight time. Lighter ions travel faster, while heavier ions take more time. An ion trap stores ions and then ejects them for measurement. An Orbitrap gives high resolution and accurate mass measurement. Next, ions reach the detector. The detector converts ion impacts into electrical signals. Common detectors include the electron multiplier, which is highly sensitive, the Faraday cup, which gives stable current measurement, and the microchannel plate, which is useful for fast ion imaging. The vacuum system is also very important. It prevents ions from colliding with air molecules, maintains high vacuum, improves sensitivity, and ensures accurate ion travel. After detection, the data system processes the detector signals and creates the mass spectrum. In the mass spectrum, the x-axis shows M over Z values, and the y-axis shows relative abundance. The tallest peak is called the base peak. Finally, neutral molecules, or unwanted material that are not ionized, are removed as cut material. This reduces noise and improves data quality.